let me introduce this is Dr. Lisa Ryan. She is Assistant Professor of University of Florida in Infectious Disease Department and Global Medicine. Here, Professor Pranabesh Chakraborty. You see, sir, please, sir, Professor Pranabesh Chakraborty. He is my PhD mentor.
Yes.
Mr. Mukherjee and Prof. Pranabesh Chakraborty, both were my mentor, PhD mentor.
Okay. Nice to meet you.
Nice to meet you.
And here you see
nice to meet you.
Professor Dasgupta, Sibamoy Dasgupta. He is a director of engineering department of the university.
Oh wow, my dad's an engineer, so I come from a family of engineers. So anyway.
So here is the guy Prem Kumar. He's doing PhD, but he's a technical guy. He's helping on the webinar. Okay, this is Dr. Diamond, Gill Diamond. He's currently he's a professor in the Department of Immunology and Infectious Disease, School of Dentistry, University of Louisville, Kentucky, US. Just let me introduce our Honorable Vice-chancellor of the university. He just joined, Professor Saikat Maitra. Okay, he's Honorable Vice-chancellor, like the President of US University, you know. So
nice to meet you.
It's an honor, sir.
Yeah. It's a pleasure and honor for me too to meet you, Dr. Lisa and Dr. Gill Diamond. Very happy to see you there.
Well, thanks.
Happy to be here.
Thanks. We just moved to Kentucky from Florida, so we're all
absolutely. From my university, it's my privilege to welcome you to this webinar, and we are really grateful that you have responded to our call and agreed to share your knowledge and wisdom for the benefit of our large number of students. This I can tell you it's an overwhelming response. And here we are large number of students, only as you will be speaking. So they have gathered. This, I believe, it will be a new way of our teaching learning. And at this moment, absolutely, it will be a new way of our teaching learning. Confined at home, this is the best possible way we can do to take progress. This teaching and learning process of all students.
Well, that's it's really progressive, I think, and it shows that India is really progressive. You know, I also work for Reckitt Benckiser, which is a large company, and they are based in England, and also have a large contingent in Gurgaon, India. And I was saying to Masoom, you know, I could give my research to your students, and he's like, that's a great idea, and that's how this got started. So
this is Dr. Diamond, Gill Diamond. He is a professor in the Department of Immunology and Infectious Disease and in the School of Medicine, University of Louisville, Kentucky. Yeah.
Dentistry.
School of Dentistry.
Yeah, and Dr. Lisa Ryan is Assistant Professor in Infectious Disease and Global Medicine in University of Florida.
Yeah, I'd like to share one of my personal information. In 2010, I happened to be at Florida for some time, and that is Florida International University. Probably they know.
Miami.
Yeah.
FIU.
In Miami, yeah.
And I spent some time on Professor Dr. Choi there.
I still am affiliated with Florida.
So before starting the speech, we will inform some rules and regulation of webinar to the participants. You know, to the participants, we will inform some rules and regulations. After that, our Vice-chancellor, Honorable Vice-chancellor, will deliver his keynote address. Then Dr. Lisa Ryan will give her speech. Then question and answers session for Dr. Lisa Ryan's speech. Then you will start. Okay.
Okay. Great.
Total registration 2,153. 2153.
Wow.
Yeah. Start it.
Good evening, everybody. It's a huge participants right now. I am Dr. M Hossain, Head of the Department Pharmaceutical Technology, Maulana Abul Kalam Azad University of Technology, shortly Makaut, Bengal, India. I am the moderator of the webinar today. I heartily welcome you all today in this particular webinar. It's a huge participants. We are highly delighted to have you all in this particular webinar. So topics of the webinar today is the innate immunity to respiratory invasion. This is very significant title in present scenario of the COVID-19 crisis throughout the world. So the title is very relevant topic in the current scenario, definitely. So without wasting valuable time, let me introduce the eminent speakers with you today. We have with us in this webinar. So we have with us Professor Saikat Maitra, Honorable Vice-chancellor of Maulana Abul Kalam Azad University of Technology, Makaut, West Bengal. This is the largest university of West Bengal. Professor Maitra is a versatile talent and eminent personality in our state as well as in our country. So here we have also with us Dr. Lisa Catherine Ryan. She is assistant professor in the Department of Infectious Disease and Global Medicine, University of Florida, USA. And we have with us Dr. Gill Diamond, Professor Infectious Disease and Immunology, University of Louisville, USA. It's really exciting to have all of you here today. We have with us Professor Pranabesh Chakraborty. He is a stalwart person in the field of pharmaceutical technology. Professor Shivamay Dasgupta, the Director of Engineering Science in our university. And we have with us our host, he's very excellent guy, Prem Kumar. He helps a lot to organize this webinar. He's with us. He's a host right now. It's really exciting to have all of you here today. Let me give you important rules to my dear participants. Under any circumstances, participants are not allowed to unmute the microphone. Please keep your microphone mute. The duration of webinar is 2 hours. We are recording the session and we will make available to you in our university website. There will be 10 minutes question and answer session after lecture of each speaker. So for any questions, participants are suggested to send their questions to the chat box. Since many people are today, we may not be able to answer all the questions, but we will try to pick up some representative questions to cover most of the queries. After the webinar, you will receive a link in your mail containing MCQ question, which is a quiz of the webinar. You have to answer it and submit for evaluation. If you get satisfactory marks in evaluation, you will be able to download your e-certificate, that is digital certificates and lecture of the PPT. Finally, I would request the participants to post or upload the e-certificates and say few lines about Makaut webinar in the Instagram, LinkedIn, Facebook page of yours. And for learned speakers, I would like to just mention that the duration of lecture of each speaker is 40 minutes. At the end, the speech will be summed up by the Professor Pranabesh Chakraborty. So without wasting valuable time, I would like to request our Honorable Vice-chancellor, Maulana Abul Kalam Azad University of Technology, to deliver keynote address of this program. Yeah, please.
Well, it's great experience for all of us. We never thought that our teaching learning process will take this dimension. And there's a huge number of participants and learners from this side, this country. They will be connected to professors from US. I'm very happy that our head of the Department of Pharmaceutical Technology, Dr. Hossain, took the initiative in making this happen. And all these experts and faculty members and technical support team for these webinars, I would like to congratulate them for making this a gala success. And it's my sincerest thanks to Professor Lisa Ryan and Professor Gill Diamond for responding to our call to give their important deliberation on an important topic in the present context. being the head of this university, it's a customary measure for me to say something about this topic, innate immunity against respiratory invasion. I would like to take a few minutes only just to give a summarized view in this way that inhalation is the lifetime mandate during our respiration. We all know earlier many people, they died due to SARS, MERS, and Ebola viral infection. Pneumonia remains the fifth cause of death in children and common cause of hospital admission in adults. Currently, we are facing the pandemic COVID-19 worldwide. This is highly contagious and fatal. So viruses are becoming a great life threat in this world right now. The lung is a battle zone. In every breath, we are exposed to environmental pathogen. Lung has direct contact with the outside world. It is exposed to so many pathogens and hazardous and toxic particles. To fight such infection, our lung depends on our innate immunity. This is basically a natural immunity. Now, the immunity are of two types. One is innate, another is adaptive. Innate immunity system, this is the first line of defense against these invading pathogens. It is a defense system with which we are born. It comes within an hour after the antigen appears in the body. Cough reflex, enzyme in tears, skin oil, mucus, which trap virus, bacteria, and small particles. These are all the common examples of innate immunity. Adaptive immunity, on the other hand, it actually develops after vaccination. And this is basically passive immunity. The innate immunity, this renders protection by epithelial barrier and mucus production. And this is supported by T cells and phagocytic cells. These cells trigger the release of cytokines, chemokines, and host defense antimicrobial to scavenge the pathogenic organism from our body. It is therefore a need of this hour. We understand this genotype of this virus, mechanism of pathogenesis, immune response, and develop vaccine and antiviral drugs to minimize infection and promote well-being. This topic today is rightfully selected, will be deliberated by two main experts in this field, and I believe that all the participants, they will be immensely benefited from these deliberations. We have introduced some new features to webinars, which has been explained by Dr. Hossain. You will be issued certificates. This is one of the new things. And there will be a discussion forum probably after this webinar is over, so that your curiosity, your interaction, that can be continued with the experts, with the formation of a community for generation of knowledge further in this field. Thank you all.
Thank you, sir. Thank you, sir. Thank you so much. Thank you so much. Thank you, Professor Saikat Maitra, our Honorable Vice-chancellor of our university, for wonderful speech. Now, without wasting time, I would like to request Dr. Lisa Catherine Ryan. She is assistant professor, she is a scientist, she is a professor in infectious disease department and global medicine department in the University of Florida, United States. Lisa Ryan, she is a toxicologist basically. She is alumni of Harvard Medical School She did postdoc in Harvard University. So without much giving the production of Lisha Ryan, I would like to request her to deliver her speech. Her topics, uh, her topics is the innate immune system in the lung and its relation to respiratory viral infection.
Hello, good morning. So I'm going to share my screen now. Is that right? Good? Okay,
Yeah.
everybody can see. Well, good morning from Louisville, Kentucky. I'm currently, but I'm currently assistant professor at the University of Florida. I'm quarantining in Louisville. The innate immune system in the lung is, as you were told, is very important. And I'd first like to thank Dr. Masoom Chowdhury and the Makaut West Bengal University and the organizers for inviting me to this webinar. It's an honor to be here, even though I'm not actually there. Today, given the urgency of COVID-19, I will speak about the innate immune system in the lung and its response to influenza infection and try to make relationships with COVID-19. And I will present mostly in vivo data. I have a mouse model focusing on the pathogenesis of influenza. There's not a lot of molecular biology or virology in here. So I just wanted to use this model, and hopefully the principles of my model will give ideas about therapy for COVID-19 and manipulation of the innate immune system to understand how the innate immune system can be manipulated and how it can modulate the outcome of the infection using outside factors. So first of all, I'd like to, let's see, I'm trying to, okay, I got it. This is my first Zoom seminar, so bear with me. I'd like to talk about the upper airways. There's a tracheoepithelium and bronchioepithelium. So when a virus droplet, a lot of viruses are in droplets. So when somebody coughs, there's a droplet. If it's large, it will embed in the upper airway, in the trachea, and possibly the bronchi. The larger the particle, the further up in the airway it will land as you breathe in. The smaller the particle, the deeper in the lung. So particles about 2.5 micrometers or less will go deep into the lung, and particles about 10 micrometers or more will go into the trachea and the bronchi. So with this in mind, where the particle lands and what it encounters is important. So in the upper airways, we have a system where we can get rid of particles, and you all probably know this, but there are, in B, that you can see that there is a diagram of the airway epithelium, and that the mucin layer here coats the airway epithelial cells, and there are cilia that beats this, moves this mucin layer, this mucus layer forward. And this is produced by the goblet cells, and underneath there are various cells that are producing cytokines and things like that. So each cell is important for as you see this list. I'm not going to read it. You can look at it later. Cytokines, mediators, antimicrobial peptides, and so on, which immediately react with the pathogen and try to either kill it or activate other cells to come in and kill it. So, and this is a C and D are stains of the airway epithelium that you can look at, and we see that this is a protein called MUC5B, and it basically is a mucin molecule that is very important in innate immunity in the upper airway. Lower airways, if a particle goes downward, and in the case of viral infection, it might land in the trachea, but then as it infects the epithelial cells and goes down into the lung, it meets a different set of epithelial cells. There are type 1 and type 2 epithelial cells, and the importance of the alveoli is that's where the gas exchange occurs. Oxygen passes through the alveoli and goes to the capillary and to the cardiovascular system. And everybody who's had physiology knows this, but just to point out this. And another, so in the lung, the deep lung is supposed to be sterile, although they are currently sequencing the microbiome of the lung. But there's alveolar macrophages, and these are responsible for, when activated, can also produce cytokines and mediators that are part of the innate immune system. They can, and I'll show you a diagram of that, but these are epithelial cells and some of the surfactant proteins that that are involved in innate immunity and keeping the lung sterile, relatively sterile. So every cell in the lung has a system where it can recognize patterned molecules like repeating chemicals, repeating sugars, repeating nucleic acids that trigger something called the toll-like receptors. And here we have 11 toll-like receptors, and each one of these receptors is responsible for recognizing pathogenic patterned molecules in the surface of bacteria, viruses, and fungi. And so one of the particular for flu is TLR3. It recognizes double-stranded RNA, and influenza and COVID are RNA viruses. Herpes viruses, in contrast, which are DNA viruses, they will trigger TLR9, and Dr. Diamond will talk about those. So not only do we have a mechanical system, we have antimicrobial peptides and mediators killing bacteria and viruses, and we also have a system that recognizes some of the patterns on these molecules, on these pathogens. Now, I just want to point out to all the toxicologists that TLR4 can also be triggered by ozone, and also anything that's a repeating molecule can trigger an inflammatory response through a signaling pathway that releases inflammatory mediators. But I will not talk about that today. Innate immune and adaptive immunity interplay with each other. First, innate immunity occurs in the lung, and like I said, and these cells down here are what we used to call innate immunity when you think about it, but the role of the epithelial cell is really important. So the epithelial cytokines, which are released by triggering these TLRs, cause a signaling pathway here, and here's the double-stranded RNA, and basically it releases a bunch of cytokines, chemokines, interferons, and many of these cytokines and chemokines call in some of the some of the cells which can immediately react and clear the pathogen for the lung. In the case of viruses, CD8 T cells are important for lung clearance later on, but immediately you get a granulocyte and NK response, natural killer cells. They're not here, but, and also the dendritic cell is important in recognizing the antigen and linking the innate with the adaptive immune system. And the T and B cells are part of these CD4 T cells and B cells are part of the adaptive immune system, which make the antibodies. And once you have antibodies, ideally, they will react with the virus and neutralize it. But we don't know that about COVID-19 yet. So, but influenza, yes, a vaccine takes care of it right away with the antibodies you've formed. The innate immunity in the airway epithelium is, these are some of the cytokines and host defense genes that are activated. Today I will talk about one of the beta defensins and reactive nitrogen and oxygen species, which are play a big role in activating other things. So this is a big network and very complex. A little bit about COVID-19. I looked this up on May 7th, and in a 2-month period, over 73,000 have died from COVID-19 infection, and 1.2 million cases in the United States have occurred. Respiratory infection it's mostly respiratory, but lately it appears it's coming out as cases and stuff, and there were a few papers out recently about the deleterious effect on the cardiovascular system. And it could explain why certain populations are affected with this virus in a more adverse way. High proportion of asymptomatic carriers. This is the bane of our existence, why we are, it's one reason why we are quarantining is because, and just as an example, 18% on the Japanese cruise ship that we heard about in the news were without symptoms out of 634 positive tests on board. So it's quite high. Transmitted, as I said, it's transmitted through respiratory droplets, human contact, and on surfaces of inanimate objects. So it's very contagious, but it's less contagious than measles, which is like if you walk into a room with someone with measles, you'll get it. It can linger for days, but it's killed with direct sunlight. It's also killed with 1 to 10% bleach and other biocides like 70% alcohol, for example. But what's most effective, oddly enough, is soap and water because it disrupts the viral membrane envelope. So it shares similarities with influenza virus and SARS and MERS in terms of its pathogenesis, and but it has some extra features that we are still studying. The latest being the autoimmune reaction and rash and cardiovascular system reactions that are like Kawasaki disease in children. We didn't think children were affected, but a rare subset of the population is starting to be affected in children. So there's a public health warning out for that. In the US, everybody says, well, it's just like flu. Well, at the minimum, 10,000 people in a year in the US die from flu, and there's about 100,000 dying all over the world per year. We're fast approaching those numbers, and we've exceeded that in 2 months with COVID. So we also, there's also a concern with bioterrorism, and we wondered if the viral genome can be manipulated and yield a more virulent strain. And so with this concern, it's hard to keep up with vaccines and stuff. So a little bit more about influenza virus. Like COVID, it's a double-stranded RNA virus with a lipid envelope. I think one's negative and one's positive strand, but basically it's an RNA virus. And it infects alveolar macrophages and epithelial cells in the respiratory tract, and it's classified as influenza A or B, and it's classified by the hemagglutinin and neuraminidase glycoproteins on the virus. H1N1 and H3N2 are most commonly studied. Our mouse model, which we published on, is in these 5 papers. I'm going to present data from 4 of them, and the 5th one you can read on your own. I want to illustrate the utility of this mouse animal model because it's quite pathogenic, and it was created in the U.S. Environmental Protection Agency by Gary Burleson. And what he did was he used Hong Kong 1968 influenza A virus, which is a H3N2 strain. And he did 18 serial infections in mice and came up with a mouse adapted strain, which has high mortality in mice, but 0 in humans. You can literally work with this on the bench. And there's, to give you an example, 300 plaque forming units is how we count virus. It's a plaquing virus, influenza is. It's an LD40, which is around 40% mortality if 10 mice are infected. So the way it's dosed is an intranasal infection, and we just drip it into the nose, and death occurs in 5 to 7 days. And this is prior to the development of adaptive immunity, which I said before was specific antibodies to influenza. Adaptive immunity takes 7 to 14 days to develop. So just keep that in mind as I present this data. The mouse adapted influenza virus strain has not been sequenced. So this particular one, I hope to do that someday. But samples, you can, from this model, we got bronchoalveolar lavage fluid, which is gently washing the lung with Hanks balanced salt solution and centrifuging the cells. You can get cell morphology and cytokine data from this BAL fluid. Then in separate experiments, we can do snap freeze the lung and get a lung homogenate assaying for cytokines, mediators, and indicators of lung damage. We can also find out virus titers using the MDCK plaque assay in determining the viral load of live virus, meaning how many viruses are actually in the lung in the infection at any time point. And then we can also use frozen lung sections to assay for cytokines with immunohistochemistry, and also using the blood, we can get serum to discover antibodies, cytokines, glucose, and cortisol. By just counting the animals, we can determine percent survival daily, along with weighing them in comparison to their original weight. We can determine body weight loss. So I have 4 examples. If I run out of time, I might not present the 4th, but these are very important. And my goal is to get you to realize how the innate immune system can modulate the outcome of influenza infection. So example number 1 is to delete an innate immune gene that enhances, doing this enhances the pathogenesis of influenza infection. So deleting beta defensin 1, which we've shown to be increased in both plasmacytoid dendritic cells and in monocytes in humans, we thought this would be important. And so we, and also in epithelial cells, when the virus infects, the HPD1 gene, the human beta defensin 1 gene, is suppressed, maybe as a way of getting the, helping the virus overcome the natural immunity. So when we deleted mouse beta defensin in our mouse model and infected them, as I told you, then counted mice, mortality in the MBD1 knockout mouse, where we deleted the gene, it was higher than the mouse, the wild type, its counterpart. So this is percent survival. So percent survival is lower, mortality is higher, and it was significant. It was about 40% difference. So, or actually doubled, almost double. So it's 100%. Anyway, so in also body weight loss was increased, and so a lot of people oppose mortality studies, but you can also look at a mouse model of any infection and look at body weight loss, because the mice lose weight as the infection proceeds, and day 0 is the day of infection, and you can see that after 2 days the mice start losing weight, and the weight loss was much greater in the mouse that had the innate immune gene deleted. So, and you can see this in the pathology too. This mouse model has a characteristic as about day 3 or to 6, where PMNs, polymorphonuclear leukocytes, and lymphocytes influx into the lung. And normally the lung kind of looks like this. It's kind of a hive pattern, beehive pattern, and you have a honeycomb pattern. But you can see that right here in this bronchial that there's a lot of cells influxing into the lung, and this is in the normal mouse. Now over here in D, you can see that this is the knockout mouse, and there's much more. You can practically, you don't even see where there's air exchange. It should look like a honeycomb, but it's practically filled with inflammatory cells. So we did also histological scoring, and the highlights were the inflammatory cells I told you about, and there was greater amounts of perivascular edema and greater amounts of hemorrhagic lung. The lungs literally look red when this gene is deleted. So this is published data in the Journal of Leukocyte Biology. You can look at it critically, but the conclusions are that when you delete a gene that's part of the innate immune system, the inflammation from influenza virus infection in mice increases, and you have a higher morbidity and mortality. So when we looked at MBD1 in humans, we found that different people had different levels of HPD1 in their immune cells. So we don't know what exactly the role is of beta defensin 1, but we're sure that it is important in viral infection. That has to be further investigated. Now, example number 2 is a little bit easier to understand and has more data. It was the result of a collaboration I did when I was at the EPA, the Environmental Protection Agency, and we partnered with Duke University to look at how oxygen and reactive oxygen and reactive nitrogen species affected the influenza pathogenesis. So that's our question. we used something different. We used a transgenic mouse. Now, Rodney Foltz, Dr. Rodney Foltz, who's now at, I think, the Cleveland Clinic, he had a mouse that was overproducing extracellular superoxide dismutase. Now, what's that? In the generation and degradation of ROS, there's a respiratory burst, and extracellular superoxide dismutase and other superoxide dismutases, otherwise known as SOD, and in conjunction with catalase, takes oxygen radicals, which are highly damaging to tissue, and these occur in a viral infection, and it immediately shuts it down by converting it in this manner to oxygen and water. So ECSOD is the predominant antioxidant enzyme found in the extracellular compartments. It is expressed primarily in the deep lung by the alveolar type 2 epithelial cells, and it protects against hyperoxia-induced lung injury. So it's been shown, and it may diminish the pathological effects of oxidative stress. Actually, it does, and I'll show you how. So we have markers of lung injury. So in the ECSOD mouse, which overproduces ECSOD, that's indicated by the black. So total protein in the lung lavage fluid is an indicator of damage, and so is lactate dehydrogenase. And so these are markers of injury. And you can see that the injury in the transgenic mouse is significantly less as the infection goes on and damage is done on days 5 and 7. Influenza-induced thromboxane B2 is another, which indicates vascular permeability. This is when blood vessels in the lung are damaged. That was also blunted too. You can see that it's almost twice as much in the wild type mice as it is in the transgenic mice, which have extra ECSOD in them because it's transgenic, it's overproducing. It's not a knockout, it's overproducing. So in another evidence is there was increased levels of antioxidants. You know, comparing just SOD in the mice, you could see that the transgenic indeed is producing more of the antioxidant enzyme as it's supposed to than the wild type. And that another indicator of oxidative stress, glutathione, as the infection goes on, there's increased levels of reduced glutathione in the BAL fluid in the transgenic mice. So one really simple way of looking at how an infection is affecting your model is to just weigh the lung and compare the wet and dry weight. And in the transgenic mouse, it takes a lot, the lung takes a lot less fluid during the infection. One of the hallmarks of influenza infection is that the lung takes a lot of water, and so and then in the end, after after 7 days or so, if the mouse lives, there is bacterial infection that occurs, and then pneumonia occurs, and that's just like the humans. So for markers of oxidative stress, basically you have during the infection in the wild type mice, you have an increased level of glutathione and malondialdehyde, which are indicators of lipid peroxides, and then xanthine oxidase, which are all oxidative stress indicators. And the flu increases all of these oxidative stress indicators. And in the transgenic mice, they're significantly reduced, meaning ECSOD is basically reducing the oxygen radicals in the infection. Surprisingly, generation and degradation of reactive oxygen species, nitrogen species is also affected. So this is the system we're looking at, the nitric oxide synthetases, INOS, and other enzymes basically, reduce the L-arginine, and it's formed by citrulline and nitrogen oxide, and nitrotyrosine is formed by reaction of the nitric oxides with the tyrosine in the proteins in the lung, forming nitrotyrosine. But as this damage occurs, we can see that nitrotyrosine in the lung is also blunted, surprisingly, and with the ECSOD overexpression. Influenza upregulates inducible nitric oxide synthetase and the nitrates in the lung, but of both types of mice, but it only persists in the wild type mice. And so these are also affected by overproduction of superoxide dismutase. And surprisingly, the cytokines were also affected. 3 cytokines are very important in the pro-inflammatory, in the inflammation in the lung. So they are released as the inflammation occurs, as the infection occurs. So you can see one of these is TNF-alpha, and TNF-alpha in the wild type mice, indicated by the open circles in this line here, is much more significantly higher than the transgenic mouse. So we have a reduction of TNF, we have a reduction of IL-6, and we have a reduction of interferon gamma, which are all increased with influenza infection. MIP2 alpha is a chemokine, and that calls in the neutrophils into the lung as the infection proceeds. It's highest at day 3, and it didn't even, it really affected the ability of neutrophils to come in. But neutrophils have a edged sword. The more neutrophils in the lung, the more inflammation and the more damage one can get. So it's an production of inflammation. Some inflammation is good, but this is bad. So this flu, IL-10 also increases, and it was blunted later on in infection. But IL-4 was unaffected, which is part of, I'll show you later, as part of the adaptive immune system, and it's indicating a Th2 response. So these are just the different BAL cell counts. At day 3, you can see that the PMNs are very much decreased in the transgenic mice as compared to the wild type. And even on day 7, you have this decrease. So, and lymphocytes are about the same, but they are blunted. They have a delayed response in that scenario. So these are the histopathology of the wild type mice. These are just increased magnification. On day 3, you can see that there's an influx of inflammatory cells into the alveolar spaces in the lung. And this probably happens in COVID-19 also. And the transgenic mouse does not have, you can see the honeycomb structure, and there's very little hemorrhage as compared to compared J and L, and you'll see there's a huge difference in histopathology. So I'd like to thank, these are the people I worked with that helped make this happen, and I will not dwell on this slide. You can look at it later. I thank them all. The example number 3, I wanted to point out, there was some, I wasn't going to present this data, but there's some idea of how can antibiotics normally used to kill bacteria alter influenza disease outcome. And this is kind of analogous with the hydroxychloroquine question of whether it can, but this is not hydroxychloroquine, so don't, but the principle about this is similar. So in this scenario, we treated mice with doxycycline 3 days before infection, and then did the intranasal infection on day 0. So we had several doses of doxycycline, and doxycycline is related to tetracycline. It's very good at killing broad spectrum bacteria, gram-positive, negative bacteria. And but there were some indicating studies, you know, they again were anecdotal, that indicated that doxycycline may be modulating the immune system. And so we tried it against virus, and we found that a 60 milligrams per kilogram doxycycline was effective in reducing the body weight loss of infection. And we also found that it was kind of a dose response. So we were very encouraged by this. And then we looked at the mortality, which was all over the place. And we started with an LD20, which was meaning most of the animals survived. But the 20 milligrams per kilogram and 60 milligrams per kilogram eventually killed the mice in greater numbers than having no doxycycline at all. So we said, well, what if you give it after infection? Well, after infection was even worse. We did, we looked at doxycycline's effect on body weight loss. Basically, there was no effect. And then we looked at mortality and found that it was all over the place. In the higher doses, giving it after infection, we found that doxycycline actually enhanced mortality. So that's how doxycycline modulates the infection, and it's not in a desirable manner. So it did protect body weight loss at 1 dose, but given after infection. But given prior to infection, it did not improve the mortality from infection. So I'm pretty skeptical about hydroxychloroquine. There has to be more studies. So the example 4 about how something would modulate the host response to innate immunity is the effects of UV light. And this was just to give you an example about what the dose we're giving an MED, minimal or ethereal dose of 4, which is a moderate sunburn. It's a vacation weekend at the beach without protection. So, and I just wanted to point out that the immunosuppressive effects of UV light does not only affect light-skinned people, it affects dark-skinned people too. So be careful. Contact hypersensitivity is suppressed, delayed hypersensitivity is also suppressed, and tumor rejection in allogenic mice, meaning mice that are genetically not like each other, is suppressed. So we asked the question, does immunosuppression occur when infection is in an unradiated site? So there was an indication that it does, because tuberculosis in mice is, the immune response is suppressed, and the bacteria, the mycobacteria, disseminate all over the body. And this is part of the mechanism of how in the skin, you know, we have a Th1 and Th2 response. I won't get into that, but it's quite complicated, and there's a lot of mediators that are produced, and other cells are activated, and the innate immune system is activated through UVB light and suppressed. And this is part of the adaptive immune system, where you have a precursor cell that when it encounters antigen or it goes into a T cell expansion, and it either goes to Th1 or Th2. And those are characterized by cytokine release of different, a certain pattern of cytokines. And this is how, and UV light can affect that shift. So there's 5 ways that UV light affects viral infections. First of all, direct viral mutation. And this is how you can kill viruses. You know, you have damage to the DNA or RNA of the virus with UV light. Exposure during the viral incubation period, this is exposure of the host, leads to more severe skin rashes. And then UV response elements in the viral genome can be affected. And also viral oncogenesis can be affected because you develop human papillomavirus and induce squamous cell carcinomas on sun-exposed sites. And viral immune responses are suppressed. And one of the examples of that is in a rat HSV model. They had suppressed delayed type hypersensitivity, and that allowed for HSV to disseminate throughout the body and into the brain. And these are the details. I don't want to go over this in the interest of time, but in my mouse model, we decided to test the effects of UV light, and we were fully expecting increased virus titers, but we give UV light on a shaved back of a mouse 4 days prior to, actually 3 days prior to infection. And the infection is the same as what I just described in the other models. And we did the usual collection of all kinds of things, and they're detailed here. And we did cytokine analysis as well. So what we found was a big shock. We had, in the combination of UV and virus, we were able to show a dose response to mortality. Mortality doubled if you compare the light virus to the UV virus treatment, the highest UV virus treatment, which is equivalent to an MED of 4. You got doubled mortality. So, but remember this, the mice have never seen influenza before, so they were immunologically new to it. They didn't have, you know, circulating antibodies like we do. So the body weight loss was tremendous too. So here is where you give UV light before infection, and then you get an increased body weight loss in the UV virus group as compared to mice that were just treated with fluorescent light. If you give them both at the same time, you don't have this pathological effect. Virus titers, surprisingly, were not different. They weren't significantly different at all at any point in the infection course. So what was going on? Well, we looked at the histopathology, and we saw that with the UV light and virus, there was a huge increase in hemorrhaging and damage to the lung somehow. And here's the normal up here in the upper left, and just compare these two. With the light virus, this is a typical virus infection, and we saw hemorrhaging, but not to the extent we saw in the UV virus group. And then we were really puzzled, so we looked at the thymus. The thymus is the source of T cells. And in the UV light and virus group, the thymus was almost obliterated. Here's the normal thymus up here. And then UV had, shrunk it just a little bit. And then the influenza virus also decreases the thymus, but not like the UV plus influenza. We could hardly find the thymus sometimes. Cell morphology was the same in these groups. Basically, macrophages increase a little bit, and PMNs, as I pointed out, increase a lot. Both groups were not significantly different from each other, and except for the lymphocytes late in the infection, the UV virus infected lymphocytes were lower than the wild type. I mean, the light treated virus, the normal infection. So here's the normal infection, and here is the UV virus infected. So then we looked at the cytokines. We said, oh, well, UV light induces TNF, so does influenza. Let's look at both in context. And indeed, it did not, we didn't see any kind of difference in the UV virus group compared with influenza. So we saw just, it wasn't enough to really make any conclusions. So then we looked, I lost myself. Then we looked at IL-6, which IL-6 is follows TNF in a cytokine cascade. I mean, and IL-6 was induced late in the infection, and in the UV virus group, it was greater than anything else. And so you looked at BAL fluid and in lung homogenates, and D is the lung homogenates, and we saw an increase in IL-6. So then we looked at some of the other Th1 cytokines, because influenza initiates a Th1 response. So when we looked at interferon gamma, we saw that there was a decrease in interferon gamma, and then later in the UV virus group, but there's an increase in interferon gamma later on with the virus. So the early, I mean, that would have to be researched, but the early suppression of interferon gamma was interesting. And but in the BAL, that was the BAL fluid. This is the lung homogenates, and we saw this a similar thing. We saw in the lung homogenates, we saw an increased interferon gamma early on, and and then later on a decrease. So there was some modulation in interferon gamma and at certain time points in the study. And in some time points there was an increase early on, and then there was later on in the lung homogenates there was a decrease, probably as interferon gamma shifted to the extracellular space. But anyway, the conclusions of this study was you did get a modulation before antibodies were formed in a double virus-induced mortality. The anesthesia we used did not influence this mortality, and the UV accelerated body weight loss and mortality in virus-infected mice in a dose-dependent manner. It did not enhance virus titer. It was only found in the lung, and the virus, when we looked in all organs, and it didn't disseminate. It was just found in the lung, and it induced apoptosis in the bronchoalveolar lavage cells. We also looked at bacteria. There were no secondary bacterial infections in the lung, and UV had no effect on these counts at all. UV had no significant effect on the virus-induced macrophage and neutrophil influx, or certain cytokines, but UV suppressed the virus-induced increases in lymphocytes in the BAL in the lung 1 day prior to animal mortality. It also modulated interferon gamma and IL-6. Interferon gamma were increased during the infection, and so was IL-6, where the UV treated group was significantly higher than the light virus group. And in other parts, it was other parts of the infection, interferon gamma had dropped. So it was interesting how this occurred during the course of infection. TNF was also not significantly increased, and that's something that really surprised us. So with this, I hope that this convinced you that there are ways of modulating the innate immune response, and that therapies can be developed that influence the pathogenesis of these deadly viruses. Thank you very much. I'd like to thank the speakers. So, are there any questions? Masoom? Is everybody asleep? Hello?
Hello. Yes, so Hossain, can you invite some questions? Dr. Hossain, can you hear me?
I think he's dropped off.
Let me contact him.
Yeah, sir.
Yeah, can you invite some questions?
Yeah, already I got some questions.
Yeah.
Already.
Yeah.
Okay. Question for you. Can anyone tell the status of COVID-19 vaccine development across the globe and the time of availability vaccine to the public around the world?
I'd say,
Very common
I mean,
question.
This was
Very
not
common
a
question.
topic. Statistical.
I'm not sure I can speak to that, but from what they say on the news, there's several companies which have candidates in development, but you have to, if you're developing a vaccine, you have to do the safety studies, you have to do, and even if you waive those and kind of combine phase 1 and 2, you really have to, you know, there were people that were studying coronavirus before COVID-19 hit, and those people are a little bit ahead of everybody else. So I think Johnson & Johnson, Pfizer, and a few of the big companies are have candidates in the pipeline. And they started yesterday. They talked about doing vaccine trials in humans. So I would say, but you have to remember you have production and supply chain as well, and you need everybody in the world vaccinated. So it could be a year or so out.
Okay, thank you. Thank you. Another question, ma'am.
Yes.
This is asked by Rimi Mondol. She asked that if the viruses are kept in the minimal temperature, such as -30°, what happens to the virus? If it is kept in the high pressure and temperature, does it get denatured?
Well, I cannot speak for COVID-19, but for influenza, I keep my virus. One of the problems with influenza, or any virus, if you're studying it, is that it can mutate and or get you know, changed genetically. So when you're doing experiments, you must take your lung homogenates, which is what they are in, right? They're in mouse lung homogenates, and you have to aliquot the virus, and you store it at -80. That does not kill the virus. -80 degrees centigrade does not kill the virus. In fact, it preserves it. So in fact, influenza virus, you can freeze and thaw a virus, which normally would kill something like HIV or other label viruses. Influenza virus is quite hardy, and you know, I've done virus titers on frozen and thawed at least twice on an aliquot of frozen and thawed virus, and the virus titer has remained the same. So
okay, great. So another question I just received, that this is very interesting questions. He's asking that some people are showing asymptomatic, you know, due to the infection of COVID. Is this the result of immunity or mutation? Why people are showing asymptomatic?
I think my opinion, and I want to say that there's no data on this, is it's due to host factors, meaning how good your innate immune system is. I really believe that. So this is influenced by age, genetics, and other things. But I suspect that the cardiovascular system involvement is due to perhaps infection of the endothelial cells, which nobody's looked at. So we'll see. I mean, there's really no data. I can't even comment. There's no data.
Okay, so
there's no, I mean, we need a lot of the cellular data. I mean, we've been researching influenza for years and done all kinds of experiments, manipulating innate immune genes on the virus that activate innate immune genes, you know, manipulating the virus itself by deleting neuraminases and mutating them and seeing what happens in cellular experiments, and to try to get an idea of what is causing the pathogenesis. We haven't done those experiments with COVID to my knowledge, but right now we are just observing, and there will be a lot of research on COVID coming out in the future.
So do you think that there is a link of endothelial cell damage to the death of the patient?
I was just guessing, but yeah.
Okay.
Yes, I mean, hemorrhaged lungs are a characteristic of COVID infection.
Yeah, many patients are showing that platelet aggregation or clotting.
Yeah. Yeah, that's another thing. It can, I mean, we don't know. At least I haven't seen any data on clotting factors. I mean, I understand that clots are forming as soon as they're dissolved and things like that. But as far as the mechanism of how the virus infects what cell and what's the cascade of mediators that is produced in endothelial cells, that's not published.
Okay. Ma'am, why only doxycycline is used, not other tetracycline?
Well, doxycycline was shown in other studies to be, you know, to be effective in changing the outcome of parasitic infection, which, as you know, doxycycline does not affect that. But so we got the idea that it may modulate host responses. That's all. I mean, doxycycline is a long-acting tetracycline, I believe. So that's why.
Thank you. Now the last questions from Muhammad Yunus. Dr. Muhammad Yunus, what and how are the natural immune boosters acting in treating respiratory infections? What are the natural immune boosters?
Well, Dr. Diamond will talk about vitamin D and in depth, so I think he can answer that. Boosting immunity to virus, anything that I think antioxidants of any kind. I was trying to get a hold of ECSOD or some kind of SOD as a way to treat mice in my infection, but I couldn't because it's not long acting as a drug. So anything that would stabilize SOD or anything that would, I mean, I think antioxidants hold
the way we're going to go is basing them on innate immune defenses, and I'd like to thank Dr. Ryan for giving a good background on innate immunity, so that I won't have to give too much here. Now it's obvious we have a how do I advance this? We have a viral pandemic on our hands. So the question is, how do we stop it? And obviously the first thing that we're going to try to do is make a vaccine. And as was discussed, many people are actively working on making a vaccine, and that would really help shut this down. The second approach would be to make some sort of an antiviral drug that will kill the virus in infected individuals. Remdesivir is something that they're looking at right now. It was an antiviral drug that was developed for Ebola, which seems to possibly have some activity in infected individuals by attacking the virus in the infected cell. And the third way that I'm going to talk about is to somehow enhance our own natural antiviral defenses. So when we think about our immune defenses, as was mentioned before, we really can arbitrarily divide our immunity into innate immunity and acquired immunity, where innate immunity is the first line of defense. It's rapid, it's non-specific recognition of the pathogens. As Dr. Ryan mentioned, we can distinguish between a virus, or say a DNA virus and an RNA virus, and a gram-negative bacterium and a gram-positive bacterium, but it doesn't look at specific antigens the way the acquired immunity does with B cells and T cells and developing antibodies. But our innate immunity has many different components to it, including the phagocytic cells that were talked about, and chemokines and cytokines, complement. We also have naturally occurring bacteria and fungi in our various surfaces that also protect us, and antimicrobial peptides. And Dr. Ryan mentioned one of those, human beta defensin 1. I'm going to be talking about another one. So before we get into that, let's talk a little bit about what we're trying to kill. Here's just an example, an illustration of a virus. In this case, it's herpes simplex virus. I'll be talking a little bit about herpes simplex virus, also known as HSV. And what you can see here, so the important points to take away from this is that this is an envelope virus, just like SARS-CoV-2, the virus that causes COVID-19. It's got a phospholipid bilayer envelope around it, which is derived from the host cell membrane. So it's like our own host cell. Inside is a protein coat called the capsid around the nucleic acid. And then the next thing we have to think about is how a virus actually works, which is, in this case of HSV, because it's got this membrane envelope, it will fuse with the host cell membrane, effectively injecting the capsid into the cell where it uncoats, and the nucleic acid then leads to replication and ultimately transcription of viral messages and protein synthesis, and it recoats the virus, and then you make lots more and they bud off. Now you have a whole lot of viruses that are coated with that particular membrane. Now, if we want to make an antiviral drug, something that actually attacks the virus, something that's based on our own innate immune system, we want to think about where we're going to take it from. So I'm going to talk about antimicrobial peptides. And as we'll see, they're found in a lot of places. Here's just an illustration of another type of herpes virus that we were working on. In this case, it's Kaposi sarcoma-associated herpes virus, KSHV, also known as human herpes virus 8. And it's orally transmitted, so it goes through the saliva. And in the saliva, we happen to have naturally occurring antimicrobial proteins that will attack this. So while the virus ultimately will infect B cells and endothelial cells and some other cells, it has to get through a large innate immune defense system in our mouth before it can get there. And this is what we're going to try to take care of, utilize this innate immune defense mechanism to somehow kill the virus. So let's talk a little bit about antimicrobial peptides and what they are. Antimicrobial peptides are short proteins. They're generally in the neighborhood of 25 to 40 amino acids in length. They're usually cationic, meaning that they have a lot of arginines and lysines, which give them this cationic characteristic. When I say they're broad spectrum antimicrobials, it means that they will kill in vitro anyway, pretty much anything you throw at them. Gram-positive bacteria, gram-negative bacteria, fungi, some viruses, mycobacteria, things like that. So they are part of our innate immune defenses that will pretty much take care of anything. And how they work is by acting on microbial membranes. They will insert into the microbial membranes. First, they'll distinguish the microbial membrane from the host cell membrane, and they'll integrate into the membrane and either cause a pore or otherwise disrupt the membrane, thus killing the microorganism. They're found pretty much everywhere. Often they're found in mucosal surfaces. So, for example, in the saliva, as I mentioned. In other animals, they're found in different places. So, for example, if you pick up a fish or a frog and you find it slimy, there's antimicrobial peptides in those skin secretions. We find them in our own mucous membranes, the trachea, the small intestine, female reproductive tract. Anywhere you have a wet mucosal surface, you're going to find these antimicrobial peptides. But we also find them in the cells of our innate immune defenses, neutrophils, alveolar macrophages, things that were mentioned by Dr. Ryan before, as well as dendritic cells. You'll find these antimicrobial peptides there. Now, the ones I'll be talking about, there is actually a larger list, but the predominant ones, we can divide them into two categories, the defensins and the cathelicidins. I'm going to be talking about a cathelicidin type of an antimicrobial peptide, but I'll mention these defensins just so to give you a little background. There's effectively two types, alpha and beta defensins. Then the difference really just is a difference in their structure, but both of them are basic cationic, that is cysteine-rich antimicrobial peptides. They each have 6 cysteines in a very highly defined consensus sequence. And as I mentioned, they're broad spectrum in their antimicrobial activity. And they're found, depending on the type, either in neutrophils or the Paneth cell of the small intestine, or in epithelial cells, and a number of other cells throughout the body. Now, cathelicidin is a family of antimicrobial peptides that is found throughout all vertebrates, as far as we know. Some animals have many more than others. For example, I believe cows have quite a large number of different cathelicidins. We only have one. We have one cathelicidin antimicrobial peptide, and it's called LL37. It's called that for a very simple name. It's 37 amino acids long, and it begins with 2 leucines. So it's this 37 amino acid long peptide that we call L37 that's part of a larger molecule that's broken down into this mature peptide. It was initially found in neutrophils, but it's also been discovered in epithelial cells. And it's made as a larger precursor molecule. This precursor molecule is called HCAP18. And HCAP18 is then cleaved down by any one of a number of enzymes to this mature active LL37 molecule. And like other antimicrobial peptides, it acts by disrupting microbial membranes. It inserts into the membrane and breaks it down. In addition, what we find with LL37 is it has a lot of other activities in the body, usually involved in modulating the immune system, either by acting as a chemokine to attract certain cells or as an anti-inflammatory molecule to reduce some inflammation. So it's a multifunctional molecule, and we believe it's extremely important in our host defense against a variety of infections. So the question is, can we use, or how can we use, LL37 in an antiviral strategy? So the first step I'm going to show you is some research that was done by my graduate student, David Bryce, on how to use it as a direct inactivator of viruses. Let me just give you a little more description of cathelicidin. Here is how it's made. It's made, as I mentioned, as a larger molecule. This is the HCAP18 I talked about. It's a product of a gene called CAMP, or cathelicidin antimicrobial peptide. So here's the gene. Here is the RNA that encodes the protein, and here's the larger protein, which is cleaved down to this LL37 molecule. And it sort of looks like this. What's important about the structure is that it's an amphipathic alpha helix. where one side has all those cationic residues, so it's hydrophilic and cationic, and on the other side are the hydrophobic residues. And that's pretty much, we believe, how it inserts into the membrane, which, as you remember, membranes are these phospholipid bilayers, so it has both a hydrophilic and hydrophobic regions, and you can use this to get into the membrane. Now, to the work that, that David Bryce did, which was looking at, and the virus he chose was Kaposi sarcoma-associated herpes virus, HV8. It's what causes the tumor Kaposi sarcoma, most commonly found in patients with HIV. And as I said, it's an orally transmitted virus. And what he showed was when he added LL37 to the virus, and here we used a virus that expresses green fluorescent protein so we could follow it. Quite simply, the more virus you add, the more LL37, excuse me, the more LL37 you added to the virus outside the cell, you just added it to the virus and then infected the cells, you saw a reduction in infectivity of the virus, suggesting that somehow LL37 was killing the virus. Now, what we compared it to was scrambled LL37, which is the same exact amino acids found in LL37, but the amino acids are in a different order. So the order is actually randomly put in and scrambled so that it has the same amino acids, but it's in a different sequence. And that doesn't work. The next question is it killing it in the cell or is it killing it outside the cell? And what we showed is that if you have to actually inactivate the virus outside the cell, and it prevents internalization. Rather than it killing the virus while it's still in the cell, the LL37 is actually preventing the virus from getting into the cell and internalizing, suggesting that the activity is happening all outside of the cell, unlike antiviral drugs like remdesivir or acyclovir or things like that, which act on a virally infected cell. The question is, how is it doing this? And if we look at the membrane, and this is, these are electron micrographs taken of cryo-electron microscopy. Here is the KSHV virion, and you can see this right out here is the viral envelope. This more dense circle is the capsid, with the protein code around the nucleic acid, and this is just untreated virus. This over here on the right is virus treated with Triton X100, which is a detergent, and that completely strips away the membrane, the viral envelope, and all you see are the capsids. And when we treat the virus with LL37 for 2 hours outside of the cell, we see the envelope is torn apart. In some places, it's just disintegrated completely. All we see is just the capsid, just like this. Other places, we just get disrupted membranes. So what it's telling us is that the LL37 is acting by disrupting the viral envelope. So what we have here is a molecule that is antiviral and can actually disrupt and kill, inactivate an envelope virus similar to SARS-CoV-2. And that's why I'm using this as an example, because it's a similarly enveloped virus. And we are actively now doing those experiments. We unfortunately don't have the data yet to see if we can kill SARS-CoV-2 with LL37, but we believe that molecules like this could do that because they disrupt the viral envelope, and they're both enveloped viruses. The second question is, could we use these in some way as a therapeutic? The problem is this. The reason we could think of using something like LL37 as an antiviral therapeutic is that, well, it's naturally occurring. We always prefer things that are naturally occurring to things that we have to make, because we know that they're probably less toxic, not very toxic if it's already in our body. We know they are broad spectrum. They have lots of good activity so that it could act very well against anything we want to try to kill. Something really important about antimicrobial peptides, which I didn't talk about, is we know that, for example, with bacteria and fungi especially, but also with viruses, that as they go through a human population, they develop resistance to the drugs that we develop. It turns out because of the way they kill, antimicrobial peptides or the microbes don't develop a resistance to antimicrobial peptides. Also, they're not very antigenic, so if we do put them back into the body, we're not going to develop antibodies and not going to get rid of them. The problem is with using them as a drug, remember that these are peptides, which means they're proteins, which means they will be digested by proteases. So that's a big problem. They often tend to be expensive to produce, to make a lot, enough to give to the entire world to kill COVID-19, for example. Also, because they are rather large molecules and they have a structure that's very specific, that has charges here and other things that have structures that can be bound, they're often bound by things in the body and inactivated. So it could be problematic using it. So the question is, how do we get around this set of problems? And one area, it's a field called peptidomimetics, which I'm sure as people in the field of pharmacology you know about, where you take the structure of a peptide, figure out what's important about it, and design a small molecule that just uses the important part of that structure, the active portion of that structure, as a drug. So one area I'm going to be talking about was really pioneered by a collaborator of mine named Anneliese Barron. And it's, instead of a peptide, it's making what's called a peptoid. And a peptoid is basically an oligomer of N-substituted glycine. So whereas a normal peptide, as you all remember your peptide structure, the amino acid has a chiral carbon here, has an amide group off of one side, has another carboxyl group off of the other, and then it has the R group, all coming off of the chiral carbon. A peptoid, or an N-substituted glycine, is the R group is coming off of the nitrogen, off of the amide. And then it makes it slightly different looking, but you still have the same number of R groups also alternating on their sides, especially if you're making something like an apopathic alpha helix. That's important. But what's very important, it doesn't really have a peptide bond, so they are resistant to proteases, and they're fairly inexpensive to synthesize. So we tested some of Dr. Barron's peptoids against, in this case, we used herpes simplex virus 1. That was that first virus I showed you, which tends to cause oral lesions, and it's orally transmitted. And what we noticed, we tried 5 different peptoids at 2 different concentrations. This is one. We saw a dose-dependent reduction in the amount of virus. What we did was we treated the virus outside of the cell for, in this case, 2 hours at 37 degrees, then added the virus to the cell, waited 24 hours, and then measured how much viral DNA there was left in the cell. And we saw a definite reduction, different for each of the different peptoids. We took 2 of them. We're ignoring this one. This one we think is somewhat cytotoxic, so we don't want to use it. But the others looked good, and we saw over time, this is a time course analysis, that even within 30 minutes, we saw a reduction in the amount of virus, although at 120 minutes, we saw a very significant reduction in the amount of virus left in the cell. So what this tells us is that these peptoids could possibly be developed as antiviral drugs. They're based on LL37 structure, but they are slightly different in their structure so that they're not going to be resistant, they're not going to be sensitive to proteases, and they may be treated differently by the body, so they may last longer in the body, they may act better as antiviral drugs. And the third area I'm going to look at to use our innate immune defenses to develop antiviral drugs is asking the question, can we enhance our own cathelicidin expression to make more of it? So for that, we have to look back at the gene that encodes cathelicidin. So here's our LL37 peptide. Here's the larger propeptide molecule. And here's the gene that encodes it. It's got 4 exons. And what someone noticed a number of years back, upstream from the gene was something called a VDRE. VDRE stands for vitamin D response element. And that suggested to these investigators that vitamin D might be able to regulate the expression of cathelicidin. And if we could just simply add vitamin D to enhance cathelicidin expression, we'd have more LL37, which could, in theory, kill our viruses. So let's talk a little bit about how LL37 is induced by vitamin D. So vitamin D will induce the expression of genes through activation of a transcription factor called the vitamin D LL37 when we added the active form of vitamin D, and that's this 1,25-dihydroxy vitamin D. We'll get to that in a bit. In addition to just increasing the expression of cathelicidin of LL37, we also see that on the surface of the cells, we're able to kill bacteria. And what we did, the way we did this was by growing airway cells in a three-dimensional culture system so that they're fed from the bottom and they're sort of dry on top, just like they are in your airway, and we're able to drip a little bacteria onto the top surface, wait 3 hours, and wash them off and count how many bacteria are left. And what we see was when we added vitamin D, we saw a reduction in the number of bacteria on the surface of the cell. So that told us that by inducing the expression of LL37, we were able to increase the antibacterial activity on the surface of the cell. Similarly, if we took mice and we injected them with the active form of vitamin D, increased the amount of vitamin D in their bodies, and then infected them through the nose with either strep pneumonia or another airway pathogen, Bordetella bronchoseptica, we saw a drop in the amount of bacteria in the lungs. So together, this suggests that vitamin D is enhancing our own natural antimicrobial defenses by inducing L37. So we asked the question, how can we use this to develop vitamin D as some sort of an antimicrobial therapy for the airway? Could we, for example, just give you a lot of vitamin D, oral supplementation? Or could we deliver it topically, this active form? And since I'm always talking about the active form, why don't we just use the inactive form, which is called cholecalciferol? So to do that, I'm going to have to remind you about vitamin D, how we get vitamin D, and how it's regulated. So we always know that you can go out in the sun and get vitamin D. Some people in history are not able to do that, so there have to be other ways of getting vitamin D. So as I said, you can go into the sun, which what it will do is the UV light from the sun will get into your skin and activate 7-dehydrocholesterol, converts it to vitamin D. Or you eat something that has vitamin D, or you take a pill that has vitamin D, and that gets into your bloodstream. Now, conventional wisdom says that there is any one of a number of 25 hydroxylase enzymes that are found in the liver, which will then add a hydroxyl group to the 25 carbon, and that gives you 25 hydroxy vitamin D, which then goes back into your bloodstream to the kidney, where there's one enzyme called 1-alpha hydroxylase, which then further hydroxylates it and gives you the active form 1,25-dihydroxy vitamin D, which gets into the cell, binds to this nuclear receptor, the vitamin D receptor, which heterodimerizes with the retinoid X receptor. Ultimately, they bind to the vitamin D response element and turn on the expression of your gene. That's great. Except then first thing that happens when you activate vitamin D and it gets into a cell is the first gene that it turns on, and turns up a lot, is CYP24A1, which encodes an enzyme called vitamin D24 hydroxylase. 24 hydroxylase then hydroxylates either of these two active forms. So this is the active form and this is the almost active form, which will then lead to either of these forms, 124, 25, trihydroxy vitamin, there should be a 3 there, I apologize, trihydroxy vitamin D, or 24, 25 dihydroxy vitamin D. Both of these are inactive and will get degraded. So what your body is set up is a feedback mechanism. So when you activate vitamin D, you then inactivate it and keep the levels exactly where they should be, so you never have too much of it. So if we take a lot of vitamin D, you just eat a lot of vitamin D, or you go for a very long walk outside, it will increase your 25 hydroxy vitamin D levels, which will lead to an increase in the 24 hydroxylase, which will lead to the inactivation. And the reason for that is to prevent something called hypervitaminosis D and calcemia. It's very good to do that. The problem is it means that we can't solve our problem by taking a lot of vitamin D. Now, that said, if you're vitamin D deficient, take vitamin D and bring your levels back up. But if your blood levels of vitamin D are very good, taking a lot of vitamin D probably won't help you, because it's just your body's just going to keep the levels where it should be. So now we want to look at possibly applying vitamin D topically where it needs to go. Could we possibly apply high concentrations of vitamin D or its metabolites right to the surface of the airway? Well, if we want to put inactive vitamin D, the first thing that has to happen is it has to be activated from inactive vitamin D, from cholecalciferol to 25-hydroxy vitamin D, and from there to 125, and from that to do the activity. The problem is generally accepted that the 25-hydroxylases are in the liver and 1-alpha-hydroxylase is in the kidney. Now, there's been some research going on the past number of years and showing that certainly there's some 1-alpha-hydroxylase that's expressed in some other cells like macrophages. We wanted to see if it's expressed in the epithelial cells. And it turns out that when we looked in airway epithelial cells, we found both 25-hydroxylases and 1-alpha-hydroxylase. Right up here, this is PCR, so it shows that the 25-hydroxylase genes, these two of these, are expressed, and we actually see in a Western blot, we see the protein. This is the airway epithelial cell on the left. This is a kidney cell line on the right. It's obviously going to have this stuff in the kidney. What we find is in the airway cell, we find 1-alpha-hydroxylase and we find 2 of the 25-hydroxylases. This is a beta-active control, suggesting that we don't need to use the active form. We could just use vitamin D and it'll get converted. And what we see indeed, if we add inactive vitamin D and we take off the media from the cells, we can see an increase in the amount of 25-hydroxy vitamin D, saying that the airway epithelial cells are actually converting inactive vitamin D to the middle form, this 25 form. In addition, if we add vitamin D, just inactive vitamin D to cells, we see an induction in gene expression. It doesn't matter whether they're normal standard cultures or this 3-dimensional culture I talked about before. We can put it right on top of the cell, this 3-dimensional culture, and we get an induction of gene expression. Now, that's great in cell culture, but what happens in an animal? It turns out if we were to do, now Dr. Ryan had mentioned intranasal infection. Here we did the same concept. We dripped it into the nose and let the mice inhale the vitamin D. So it's an intranasal delivery based on the whole concept of intranasal infection. Here we used our ethanol control. It's 0.1% ethanol. And we did either vitamin D or the active form. And what we see is it didn't matter whether we used vitamin D or the active form, we still got induction of gene expression. And what that tells us is that we can actually deliver vitamin D straight into the nose of mice, and we get an induction of gene expression, which could then lead to, of course, the induction of LL37, which could, in theory, lead to the killing of whatever microbe we're talking about, potentially COVID-19 or influenza or any other envelope virus. So to summarize, LL37 can inactivate viruses outside of the cell. It's not like any of these antiviral drugs like acyclovir, which have to have the virus already infecting the cell. It occurs by disrupting the viral envelope. We could potentially mimic LL37 if we don't want to actually use the mature peptide because it may be degraded by proteases or other problems. We can mimic the structure using something called a peptoid and then give that, and we could potentially do that by maybe inhaling it and acting there. Or we could use vitamin D to induce our own expression of the LL37 gene. We know that the active form can actually do this. I didn't talk about inflammation, but it also inhibits the inflammation that we see in both flu and COVID-19 infections. The airway epithelial cell can convert inactive vitamin D to 25-hydroxy D3 as well as 125-dihydroxy D3. And then we can either add it to the apical surface of the air-liquid interface cultures or right into the airway of animals, and we can see rapid gene induction, potentially leading to a therapeutic treatment of the airway to either prevent or treat an infection. So the questions we have to ask are, can we develop LL37 as an antiviral agent to be delivered right to the airway? Could we develop these peptoids as antiviral agents also to be delivered to the airway? Or could we develop vitamin D as a therapy to induce the expression of LL37 in the airway? So in those three ways, we're attacking all three ways to see if we can use this to develop a potential antiviral therapy for treating respiratory infections. And I want to thank various people who worked on this, especially my grad student David Bryce, and a postdoc Christina DiFranco, and another postdoc Sunghan Yim who worked on it, and my technician, as well as some people who have helped out, especially Dr. Ryan, who has helped with a lot of studies, especially the herpes virus, and also showing us how to actually put things into the nose of mice, which is a very difficult technique. And my collaborator Annalise Barron at Stanford, who's developed all these peptoids, and our funding sources from the United States National Institutes of Health, as well as a company that I've been working with on the peptoids, Maxwell Biosciences. Thank you very much.
Thank you so much, Dr. Diamond, for a wonderful speech. So from audience, there are questions. Can you hear me, Dr. Diamond?
I can hear you. Yes, I just didn't want to interrupt. I figured I'd let you talk.
Okay, so question. So what is the future for prospect of LL37 as antiviral drug?
So I think the main reason I showed you this was to give a background on how LL37 works. I tend to think that LL37 will not be that good as a delivered drug because, number one, there's the potential for being digested by proteases. Number two, as far as concentrations, getting into high concentrations you probably have to deliver a lot of it, and there may be some other problems because it's immunomodulatory of it getting other places. I know there are people who are working on this, so it is quite possible that it can be developed as a drug. There are a number of labs that are working on actually developing it as a drug. I personally would like to go towards either the peptoids or the using the vitamin D as a therapy to work on that rather than using the LL37 itself, although it may actually work, and I'd like to see it.
Okay. So another questions from audience. LL37 and vitamin D can be combined to develop a therapeutic strategy against the virus.
So we're actually actively working on the vitamin D aspect. What's interesting is to support all of this that I've told you, a number of papers have just come out that have associated vitamin D levels with low vitamin D levels with poor outcome for COVID-19 patients. So it seems that if you have real severe vitamin D deficiency, you tend to do very badly if you have COVID-19 infection compared to people who are sufficient in vitamin D. So it's quite possible that this whole vitamin D thing will work out. We're actively doing the experiments now. I wish I had the data for you, but we ran into some roadblocks, not the least of which is one of the persons who was actually doing the experiment got COVID-19 and had to be the whole thing got put on hold for a couple of weeks, unfortunately. So these are the problems we're running into, but we are actively looking to see if we could develop this as an antiviral therapy.
Okay, got it. I think audience has understand. Another question is that how herd immunity, do you know herd immunity?
Sure.
Plays role to stop the pandemic virus.
Well, it would be great. As I'm not a viral immunologist. This is the problem that when you work on these things, people think that you're a viral immunologist. This is actually not what I do. So Dr. Ryan, who's actually has studied more immunology, could probably answer that better. But I believe that either we have to develop a vaccine so that a certain number, I don't know what the actual threshold is, maybe 2/3 of the population is either has been infected or has been vaccinated before we actually get to that point. Now, if we develop these therapies, these therapies sound great because they could prevent the infection, but the whole point about herd immunity is very relevant because it's great to have therapies that kill viruses, but if you're not developing herd immunity, the virus is still going to be around for a long time. So I think you need a combination of antiviral therapies as well as vaccines, and a number of people actually getting it. And hopefully, if you get it, you are actually immune. They haven't really shown that yet.
Okay. The last questions, can vitamin D can be used to treat COVID-19 virus?
So that's the question we're trying to answer. We'd love to find that out. As I said, if you are vitamin D deficient, I always recommend to anybody who asks me about vitamin D is get a blood test, find out what your vitamin D level is. If you're vitamin D deficient, go to your doctor and find out how much you should be taking, because you want to bring your vitamin D levels back up to normal. As I said, people who tend to be vitamin D deficient do worse with COVID-19. So be vitamin D deficient. If you're already sufficient, if you have the right levels, then maybe just take a minimal dose, a normal dose, like 400 to 1,000 units a day to keep it up. That is just sort of general health, and that may or may not protect you against COVID infection. We have to develop this as a therapy. The therapy we're trying to think of is actually inhaling it and getting a higher concentration directly to the airway, and we'll see if that can help, rather than taking vitamin D, which may not help at all unless you're deficient.
Great. Thank you so much. Thank you so much for your presentation and
My pleasure.
But I have a question to Dr. Ryan, Dr. Lisa Ryan.
Hello, Masoom.
Yeah. So I have a question to you. Do you think that continuous lockdown can help us to prevent from COVID-19, or do you think we go outside and face challenges to the virus and get some herd immunity? What do you think?
Well, if you look at Sweden, that's exactly what they've done. The problem with that is they're risking lives, and we don't know exactly who is we don't know enough about the virus to know who is susceptible to these really bad effects of COVID-19, and we don't know the transmission rate. There's a lot we don't know about COVID-19, and eventually we're all going to have to get it, because you can't be locked down forever. I was just thinking about this morning. However, we have to have testing in place, and we have to have the public health capabilities of treating our patients who end up getting really sick. And I think the lockdown is the right thing to do. I don't know India's numbers. It would be interesting to find that out. But in states that lock down immediately, like Kentucky, where I am, the numbers are really low, and they will open businesses and stuff very gradually with social distancing and rules like masks and washing hands and hand sanitizer. The problem with that is we have a problem getting those preventative measures in place because of the supply chain. At least here in the United States, that's the problem. So I think the lockdown was the absolute right thing to do. Getting out into the fresh air, I think without people being around you, that's probably impossible in India. But it is that's something one can do. But I honestly think that the government took the right approach. But when you get exposed to the virus, you will have to have that safety net in place.
Okay.
I'd like to actually bounce off of that if I could. Thanks for Dr. Ryan for mentioning Sweden, because if you think about it, Sweden was exactly the wrong country to test this method, because Sweden in the winter, everyone is vitamin D deficient because they're so far north, they don't get very much sunlight. So not only did they not lock down, they're also vitamin D deficient. Now, I believe there was one study that tried to look at the different latitudes, and it seems that the closer you are to more sunlight, the better you do. So the locking down is a really good idea, as long as Dr. Ryan mentioned, you get outside and get some sunlight and get some vitamin D in you. I just wanted to make that vitamin D.
You only need
also.
10 minutes.
Right. You don't need very much. You just take a walk outside for a little while.
Yeah, this is a great recommendation. Great. Okay, so
you asked a question about herd immunity.
Yeah.
Herd immunity is very important. About 70% is necessary to protect society as a whole. So I think I don't know. I don't know what the solution is. You could easily say, well, gee, let everybody be exposed and we'll just take the deaths as, I just don't think that's morally right, actually. So I think we'll have herd immunity in time. I mean, there's been people who swear that they haven't been outside and are getting it asymptomatically as we have testing going on. And remember, when you have testing for the virus, you are positive at that moment or negative at that moment. Actually, if you're positive, you theoretically you will develop antibodies. We don't know if those antibodies are protective. If you get the antibody test, you have antibodies, and that means you have at some point been infected, maybe asymptomatically. But once, as people get exposed to the virus, this herd immunity will develop, hopefully. And this is why my UV mouse study was, I thought, irrelevant. You know, I didn't really publicize that very much, because in what world are you completely completely non-exposed to a virus? We get bombarded with viruses and bacteria all the time, every day, and our innate immune system takes care of it. But if it passes that, then you have an adaptive immune response. So you have some cross-reactive antibodies against different pathogens already. And that's why I thought the UV light was irrelevant. By the way, when I forgot to mention that we compared the UV light group and the UV virus group with the light virus group in terms of antibodies. In other words, we measured antibodies and recovery and mortality based on reinfection, and we had no difference. So this UV light did not affect once the adaptive immune response was made and we made antibodies to flu, we did not see an effect of UV light on that response, that memory response. So this was strictly effects on innate immunity, and therefore irrelevant, because you have typically, except for COVID-19, you typically have at least a little bit of an antibody response or a T cell response to viruses, because you have seen them before.
Yeah. Okay. Thank you. Thank you.
I have a question to Dr. Gill Diamond. That is actually, what about the traditional medicines of different countries? Do they really help in fighting this COVID? Because many of the governments are advocating it.
So it's interesting, because I do run into a lot of papers that are out there about looking at traditional medicines. In some cases, traditional medicines actually do things like induce LL37. You do find these things that are inducing antimicrobial peptides and enhancing innate immunity. I think the research has to be done. It's very, very possible, because traditional medicines, they wouldn't have lasted this long if you didn't see some sort of an effect somewhere along the line somewhere, and they're usually based on some actual biochemical pathway that's induced, usually a gene expression, something like that. So I think, they can be looked at possibly being developed more, than just simply taking them the way they are, which is probably inefficient, but somehow finding out what the compound is and seeing if that induces LL37, for example, or some other antimicrobial peptide, or inhibiting inflammation, which is another way that they work. I think that is really good. I see a lot of it, and it seems to go places.
I have a comment, actually. I want to point out that viral load is very important. In other words, if you overwhelm the innate immune system in these antimicrobial peptides, and thinking like a pharmacologist, right? You can have it's a dose response. So you only have so much made in each cell of your epithelium, airway epithelium. So If that response is overwhelmed with an increased viral load, and this is my justification wearing a mask, those homemade cloth masks will not prevent all viruses from going through. Viruses are very small. It's like keeping mosquitoes out with a cyclone fence. It's not the same, but it will keep out a majority of the larger droplets. So you would protect yourself from getting a huge viral load, which can overwhelm the immune system in a dose-dependent way. So, and then then I think the traditional medicines and the vitamin D may not help. But under normal circumstances, I think being vitamin D deficient would be awful for you. And to keep up your vitamin D levels is very important.
Thank you. Great. I think our audience got a lot of information from Dr. Diamond and Dr. Lisa Ryan. Now, I would like to request Professor Pranabesh Chakraborty to sum up their views and findings to the participants. Please, sir. Sir, you have to unmute. Please unmute.
Can you hear me?
Yes, sir.
Okay, thank you. Very hearty congratulations, Dr. Gill and Dr. Lisa. I am starting from the inception. Honorable Vice-chancellor, Sir, Makaut, Professor Saikat Maitra, Honorable Professor Gill Diamond and Professor Lisa from USA, and Professor M Hossain, my beloved student and head of the Department of Pharmaceutical Technology. We are really thankful and delighted with your presence and thought-provoking deliberation in the webinar for our learned faculty members, respected directors and respected principals of our esteemed institutes, and beloved students of Makaut. Professor Lisa, in her lecture, depicted very nicely and lucidly the innate immunity system in the lung and its relation to respiratory viral infection. We all know that at the present juncture of our country, as well as the entire globe, is suffering from COVID-19, and a lot of burning questions are arising every nook and corner and in the minds of the most of the people. What it is, what are the sources of this killing virus, how it is spreading, how to save the mankind from this horrible and unwanted situation, etc., etc. And Professor Lisa, by their hard and painstaking research work, gathered broad spectrum knowledge and very neatly tried to make us understand about the whole concept of world of virus and immunity with a crystal clear ideas regarding the exhaustive manner. Professor Gill has demonstrated each and every tidbit of the subject. Thanks and warm regards to Professor Gill for your unique deliberation, which has positively enhanced the scientific thoughts of our students. Thank you. One question is there that nowadays it has been told by so many scientists in the globe that India has the natural immunity for two things. One is vitamin D, plenty of vitamin D as the temperature and sun rises from the early morning to evening. From dawn to dusk, all the people are getting vitamin D, one. Second, as in the childhood, they used to get BCG vaccine. That's why the infection is much more little than so many countries. What is the basis of that, particularly for BCG vaccine? Dr. Gill.
You know, I read about the BCG issue. Someone had posted that as a suggested way that it could also being vaccinated with BCG may affect COVID infection. I don't know much about it. All I do know, I did look this up. BCG does induce LL37, so it could be a case of BCG inducing LL37, but that would have to really only work at the time that it's happened, not many years later. I'd also read something about a genetic microRNA that was common in the Indian population, which may be associated with less severe COVID-19 infections. That's another thing to look out. I think as this infection goes through the world, we're going to find out things like whether BCG does provide some partial protection, whether there are genetic predispositions, so some populations may be more susceptible than others for a variety of reasons, and that will really help us develop new drugs.
Thank you, Dr. Gill. It is over to Masoom.
Thank you, Professor Chakraborty, for excellently summing up the topics of Dr. Diamond and Dr. Lisa. Before saying the vote of thanks, I would like to request Professor Saikat Maitra, Honorable Vice-chancellor, to say something.
It was a nice and a vivid deliberation. I was just noting these messages shared by many participants, and I think all of them got immensely benefited out of these two deliberations, and we will take it forward. That's why I was asking to create a discussion forum where all the participants can put their comments and their opinions and their queries. That is one of the this important takeaway of this event. And then we believe that in near future we will be inviting Professor Gill and Professor Lisa again and again for enriching ourselves from their wisdom, from their knowledge. And I wish our this webinar team a grand success in our all endeavors.
Thank you so much, sir. Thank you so much.
Thank you very much.
Thank you so much. So,
Thank you very much.
it's an immense pleasure for us to have you, both of you, with us. So, really, it's a great evening for us. On behalf of Maulana Abul Kalam Azad University of Technology, I would like to thank Dr. Lisa Dayan and Dr. Diamond for spending their valuable time with us today and delivering excellent presentation with lots of research work and nice information to the audience who are the students, faculties and scientists. I wholeheartedly thank all the attendees, participants, my friends, for taking time to attend to the webinar in a large number of participants and making it a grand success event. I would like to thank our Honorable Vice-chancellor for approving this webinar to conduct in such a way. Dr. Maitra, he just told us, all the HOD and director, to contact with the Pharma University and conduct some seminar. I tried, and I'm very happy today that sir has approved this webinar. Thanks to webinar committee. In committee, Professor Indrani Mukherjee. I'd like to thank Professor Indrani Mukherjee, the chairman of the committee. Professor Shivamoy Dasgupta, he is already here in the panel. I would like to thank Professor Somdatta Chakraborty. I'd like to thank Sujit Mukherjee, our webinar committee. Definitely Manoj Ghosh, he is also here co-host. And last but not the least, the excellent guy, the Prem Kumar, he is now the host. He has done excellent work for this conducting this webinar. I would like to thank my faculty of the Department of Pharmaceutical Technology, Professor Arup Mukherjee, Dr. Subhashish Dar, Dr. Sonia Kundu. I would like to thank all the HOD and directors and all faculties of my university who has extended our help and support to make this event a grand success. So, again, I would thank Professor Pranabesh Chakraborty, because he has done a great job for summing up this excellent lecture. It's really hard thing to sum up all these slides, because Dr. Lisa has lots of information. Dr. Diamond's also given lots of information. It's really hard to sum up it, but he did it. Thank you, sir. And thank you being a important part of this seminar. Professor Dasgupta, Srimay Dasgupta, untiringly is listening, being as a panel. Thank you so much, sir. Again, I would like to thank again to the technical person, Prem Kumar and Maitra sir. Thanks to all participants and audience. I'm sure all audience and participants have enriched their knowledge today. And in future, definitely we'll be connected each other for sharing our knowledge and information. Hope to meet you again. Thank you all.